Nerd alert! Nerd alert!
You're listening to Nerd Alert, the podcast of the Alabama School of Math and Science, and we're very excited today to talk about ocean acidification. So we have the Research Fellows Program here at ASMS, and all this year we had a big project that was very special because it was funded actually by a NOAA OAP mini grant. So we have lots of different kinds of projects that we work on in Research Fellows. This one was a very special group project, and the principal investigators are here, or as I call them, are my colleagues. Professor Natalie Ortell and Rebecca Domangue are here. But first, apparently we have a very burning question.
Yes.
It's a very serious topic that we need to go over. So please tell us this important information right away.
Why does an oyster not share its pearl?
Oh,
I don't know why.
Because it's shellfish.
Okay, thank you. So now that we've set a tone of academic rigor
Yes, absolutely.
and seriousness, I do legitimately want to hear more about some of the important lessons that our students not just learned for themselves, but were able to share with the community at our Spring Research Forum that we just had in May.
Yes, awesome. So yeah, we were really pleased and excited to have received this NOAA OAP grant to allow us to do some really deep dive into the impacts of ocean acidification. And the really cool thing is that ASMS pulls from a student body that is diverse and across the entire state, but many of them have never actually maybe even seen the ocean or know even what an oyster is or what ocean acidification is. So we were really able to bridge some missing gaps and really hit on their ocean literacy. And it's going to be really cool for everyone to see, or if they did get to see at the research forum, really how they built that ocean literacy, and then to be able to talk on the podcast and really kind of dive into that is going to be really cool.
I like how you said dive repeatedly because of the ocean.
You get it?
Dove into the ocean. Yes, definitely. What stood out for you, Dr. Domangue?
Today you'll hear from our research students on their experiences with the project from start to finish. The things that stood out for me were their tenacity, their curiosity, their ability to pivot, their ability to engineer and be creative within the boundaries of understanding a big global problem and figure out a little slice of that here locally and how that applies to Mobile Bay.
And that was why it was particularly gratifying to have such great attendance at the Spring Research Forum.
Yes.
And, we have poster sessions and we have presentations, and I know your room was packed out with not just students.
Standing room only.
Yes, we had parents come and community members come. So I think this year was definitely our best in terms of attendance at the Spring Research Forum, just moving beyond just our campus, which is great, into the community more. So it was really great to spread ocean literacy to lots of different community members. So the real stars here, of course, are the students.
Yes.
So we're going to go ahead, unless we have any more oyster jokes.
No more oyster jokes.
Okay, that's all for now. You're okay, good.
We're clammed up.
You've hit your, oh no.
That's a good one.
Oh dear. Okay. Well, having said that, we're going to try to gather what shred of dignity we have left and turn it over to our guest host, Lillian Abernathy.
Hey everyone, so first things first, we're going to all introduce ourselves. So again, I'm Lillian Abernathy, and I'll be asking some questions and maybe popping into answers a little bit. And then we have...
Hi, my name is Hyerin.
Hi, my name's Emma Kate.
I'm Naria Khristoforova.
I'm BoKyeong.
And I'm Kayty Phan.
Okay, so now that you've met everybody, so our first question is, can someone explain to me, what is ocean acidification?
Yeah, okay, so OA, or acidification, is chemically, it's when carbonic acid mixes with hydrogen ions and makes carbonic acid in our oceans, but that's what's making them acidic, right? So the pH level at a normal in our ocean needs to kind of be anywhere between 7.8 to 8. Really awesome, life happens. But under OA conditions, the pH is increasingly getting lower. And even if you see graphs, oh, it's 7.6, oh, it's 7.4, that may not seem like a big jump, but to the ocean and their ecosystems, that's a really big jump. Even the slightest decimal point can change so much for them, which is why we decided to focus our attention on that.
And what is y'all's research, or our research, actually about? What are we actually testing and quantifying in our research?
So we're looking into OA's effects on specifically oysters And oysters are a big part of the Mobile community since like we live here right next to the Gulf, and the seafood economy is really big here with restaurants as well as the oysters being exported to other areas as well.
Yeah, and specifically we looked at how OA conditions impact growth, maintenance, homeostasis, some kind of like the most key biological processes that they need, and how OA is hindering them.
Was there anything interesting that y'all found?
I'm sure you guys found lots of interesting things.
Oh my
I guess,
gosh, yeah.
what was the biggest surprise that you uncovered in your work? Tell me more about Tank 2.
Tank 2 was weird, I think, from the beginning, right? I mean, like, we had 6 tanks based on each individual projects and what they were looking at. And so Tank 2, I mean, we started taking morphometric data really early, and Tank 2 was just weird in all of those. But also, as we started to kind of study it, we noticed they got really, really sick, and none of the other 5 tanks had that same issue. And we found out they had a pathogen, and Tank 2 was the only one that had it, even though we had randomly assorted the oysters. So Tank 2 just kind of gave us a run for our money there on resilience.
Can you girls tell me more about how you divided the work up? Because you all worked on ocean acidification, but it seems like you each did your own angle on that. So maybe we can just go around the room, Lillian, we start, and then we'll go in a circle and just talk about which aspect you addressed.
So I focused on the growth aspect of oysters, and more specifically, I studied these 4 calcium-binding proteins that are found in them, and basically their goal is to capture calcium and move it throughout the oysters so that they can actually grow and expand their shells. And I was studying the effects of those proteins under ocean acidification, and I was hoping to see that when those oysters were stressed due to OA, that that protein expression would increase. And under some of them, we actually did see a little bit of that, but.
Yeah, my research mostly focused on kind of the susceptibility of oysters under ocean acidification, and I mainly looked at a marine pathogen called Parkinson's marinus, which is a really common pathogen down here in the Gulf. And I really wanted to see a decreasing susceptibility of Parkinson's marinus under ocean acidification condition, but my preliminary data didn't really show that, and it kind of suggests that even under ocean acidification, that Parkinson's marinus is still a viable pathogen for the oysters.
Nice. Yeah, so I looked at the impact of ocean acidification on specifically the nitrogen cycle. The oysters were kind of brought in because those are a big part of how that functions here. Oysters are just kind of that front runner for how that works in marine ecosystems. And so in the oyster specifically, there is a cavity called the ectopalo cavity, and they have a really unique microbiome there, as well as unique gene expression. And so in that cavity, there are 3 key genes corresponding to nitrogen fixation, nitrification, and denitrification. And so for this year, I looked at the nitrogen fixation gene, NIFH, against OA to see if there was any kind of significant change in the expression without OA conditions to what it is. And while we didn't find anything significant quite yet, I'm going to continue and optimize next year and look at 2 more genes for the other 2 parts of the process, as well as ammonium concentrations.
For me, my research was kind of branching off of Hyerin's, and where I looked for a solution to the oyster's susceptibility to pathogens. And my solution was using submerged aquatic vegetation, which is where I looked at specifically this red algae and their phytochemicals, which were fluridicide, digenicide, and succinic acid, to see if they would possibly reduce the amount of pathogenicity they have. And we didn't really see significant data to that with our preliminary data. But if we keep on going on through with our research, then we could possibly see something.
I was looking at the homeostasis aspects of the oysters. So oysters go through apoptosis to maintain their homeostasis, and I was looking at the caspase 7, which is an enzyme that plays an important role in apoptosis, and I was kind of seeing if those caspase 7 gene expressions will increase or decrease under the condition of ocean acidification, Parkinson's marinus, and acidic phytochemicals. but my preliminary data didn't really tell anything about it, so further optimization is needed for further research in the future.
And then for my research, I also looked into growth, just like Lillian. But however, for me, I'm looking into a protein that's more of a shell building protein. So it helps with the building of the shell, which, as we know, OA affects the shell because it's decreasing the calcium carbonate ions in the ocean. So I'm looking at the expression of this gene to see whether it increased or decreased because of OA. And then with my results, my preliminary data did show that there were indication that we were successfully able to extract our tissue. However, it did not cross our threshold to get a CT value when completing qPCR. So next year I will be optimizing to be able to get better values.
Yeah. And thinking about having next year too, I think for those of us who are juniors especially, we have so much more time to just focus on extractions, analysis, and optimization, whereas this year we had to spend probably a good 4 months on just building our project. We have that now. So next year, we'll probably be able to get through a lot more progress with our each individual sections.
And so for our experiments, we all used the qPCR to get our data. So can someone explain like what the qPCR is and how did we use that to actually get our data?
Yeah, so qPCR, I kind of reference it as kind of like a magnifying glass, that and the primer. So qPCR, you have to put what are called primers into your sample, and you put your sample into the machine. It's a big, like, kind of blocky machine, and it hooks up to a computer. And so the primers tell the qPCR what to look for, hence the magnifying glass analogy. And so when you hook it up to the qPCR, it's going to show you an amplification, and that's basically how much of your target is in your samples. And to interpret that, you have to look at the CT value. So the CT value, there's like a threshold, and then there's 40 cycles they have to cross that. So if, let's just say it's like a printer, if you put a sample in and it comes up really, really early, that's like there's more of it there, but if it comes up later, there's a lot less of your target there. So you would rather it come up earlier in the cycles than later, because that means you're probably going to be able to have more to support with your question.
What other new like lab skills or technology did you guys learn? Everyone's sort of laughing about that. Was it a lot? Is it too many to even list? Hyerin, what about you? What did you like? I mean, that's an exciting part about research fellows for me to hear about and not actually do myself, but is for you guys, you get exposure to stuff you haven't in the past. So Hyerin, like what would be an example of that?
Oh, well, I did definitely learn like a lot of new lab techniques. Like just like Emma Kate said, like we definitely did not know anything about qPCR at the beginning of the year, and now we're like kind of experts. But I think one lab skill or technique that I've think I really mastered this year is probably pipetting.
Yes.
Well, who else wants to tell me about pipetting? Your finest pipetting moments or other things you may have learned. Naria, everyone's smiling so knowingly. I feel like
there's a lot to pipetting. You just gotta be really careful because with our pipettes, you need to make sure you get the right dose, and also making sure that when you're holding down on the pipette, you want to make sure you don't go all the way down, or otherwise you drop your sample. So you got to be really careful. It's a technique that has to be mastered
really quickly,
especially when it comes to our research.
Yeah, and like Hyerin said, there was a lot of it, and when we say a lot, we mean a lot. There was over 300 samples, and then like a lot of our samples had to have like stages that required a lot of transfer pipetting. So we pipetted. I don't even want to guess how much we did. But I don't think there's a day we didn't do it, honestly.
It was like we did so much pipetting that we ended up naming our pipettes.
Yes, we did. Oh, yeah. Yes.
If you don't tell me the names of your pipettes right now, I'll have you all kicked out of school.
No. It was specifically 2 pipettes, and it was this little they were both like the 1,000 mil ones, and for April Fool's Day, Dr. Domangue put googly eyes on them, and one had pink googly eyes and one had blue googly eyes. And so the one with the pink googly eyes was Patricia, and the one with the blue ones was Ronaldo. Ronaldo was my favorite.
Yes, we all really often fought over those pipettes because they were the largest ones we had, and we all needed them for our protocols. And like you had to use like that dose at that like kind of like checkpoint to continue, so we had to keep waiting, and so waiting with googly-eyed pipettes made it a bit more fun.
Well, what were some other fun memories that you guys have? Everyone, it seems like it was really fun. I know there were some setbacks, like there are to all research processes. So, but I would like to hear about some of the fun things. What were some favorite memories?
We got to go to Bama Bay Oysters and actually harvest our oysters for the project and flip the cages and go in the water, which I think that might have been one of the first things we did as a group for that project, and that was really, really fun. And then we spent a lot of nights together in somebody's dorm lobby working together on things, and those just kind of turned into really fun memories because friends who do lit review together stay together.
So true. Another good memory is that one weekend where we spent opening up all our oysters.
Yes.
That was
Good times.
Yeah.
It was a couple of hours, but it was really
A couple is
fun. It
being
was
nice.
About 8 hours worth of extractions.
Well, we did end up getting all our tissues.
We did. We got it.
Yeah.
So-
I think another key moment was the sustainability conference that we went to.
Oh, that was so fun.
That was so much fun.
It was a day at the beach for science.
Tell us some more about that.
So we went down to Gulf Shores, right? The lodge.
Yeah.
And we spent the day going to the sustainability conference that was it by ACF?
Yeah, it was ACF.
ACF. And we were able to see different presentations and projects. It's like my favorite was the one about the Gulf Shores Middle School.
That was
where
fun.
They have like, their school is basically, the Gulf Shore City Schools are like on a state park.
Yeah, there you go.
Yeah, a state park. So like the students are able to like go out and during class and like learn about physical science in real time. And,
That
like,
was really
experience
cool. We listened to one about light pollution and how it corresponds to sea turtle life cycles too, which was really, really, really enjoyable actually. And I still correspond with that guy every now and then because I invited him to the symposium. Don't think he made it, but it was still fun. It was a really good opportunity, I think, for us to network because a lot of us had never had any kind of experience like this before, and so getting to kind of experience the conference without having to be presenting in the conference was really nice. I think kind of eased our nerves for a symposium, and then Kayty and I are presenting at a conference in June as well, so I'm really glad I had that opportunity to kind of see it without having to dive straight in.
Okay, so we mentioned our project and then some of the fun things and some of the stuff we learned. Now, what would we do differently or like more to kind of like optimize it, like thinking back?
Ooh, I wish we had more time.
So much more time. Yeah.
I definitely think that we need more time to do our research because we also had like some other setbacks throughout our research that we kind of
Needed to have that time for.
And then we made a lot of progress at the end of the research because the time crunch was our big of the issues that we had. And if we were to have more time to do our research, I feel like we could have gotten more in depth of it and like do more of it.
Yeah, and I think we also kind of had to rush on those extractions, which could have been some of the problem there with our preliminary data, because like we had a very clear be-all, end-all date. So I think we all kind of got stressed there towards the end too, but next year with having more time, that should be alleviated some for sure.
And so now, how does our project translate in like correspondence in like helping understand more about Mobile Bay or helping understand more about the effects of ocean acidification?
I will answer that with the question, how many restaurants can you go around here and see oysters on the half shell being advertised? I mean, anywhere you go, kind of in the near vicinity, seafood-wise, you're gonna see oysters being advertised in some capacity. And that's because the bay is in our backyard. We said that in our symposium. It really kind of is our backyard, not just in it. And oysters are such a big part of our area's economy too. So OA is a really big problem that real-time oyster farmers are facing, and they're facing them here.
Yeah, and I think kind of like to tie on to the economics effect, I do also think they do play a major role in like the ecological perspectives because their 3D structures provide a lot of habitats for other marine animals, and they're also a really good natural barrier to reduce wave energy, which could reduce shoreline erosions.
Yeah.
So what's next for all of you? You mentioned future directions, future projects, things like that. Anybody want to weigh in on that?
Oh, sadly, me and Lillian are graduating, so we won't be able to participate as much as we would want to, but we will be finishing up our papers to contribute to the research that we did.
What about after you leave ASMS? Do you think you'll take some, and not just the lessons that you've learned, but is this going to impact your college career,
Yes.
any of your choices, decisions, things like that?
For sure. I know for me personally, although I'm not pursuing a science major, but I do plan on definitely getting involved with some sustainability projects where I'm going to be going to school at, like in Columbus, Georgia. Getting involved with some volunteer work for sustainability, and also I think there's a few seafood restaurants over there, and maybe seeing if I can set up an oyster recycling program over there. I don't know how, but it would definitely be something interesting to try and do.
Yeah. I know me personally, next year I'm looking at 2 more genes. So my overall kind of overarching question was how OA impacted the overall nitrogen cycle productivity. And so this year, because of time constraints, I only looked at that first part, nitrogen fixation, but there are 2 more genes that correspond to 2 more processes that really give us that full picture. So I'm excited to dive more into those next year for sure.
Oh, so like most of our current juniors will be like working more onto research next year. So we need like further optimizer, like primers and like RNA extraction for like to get the actual CT values or like get like better CT values. And we'll be also working on a paper with Lillian and Naria even though they graduated.
Yes. To hopefully publish, maybe.
And of course, a really important question is, did you eat raw oysters before this project? Is anyone here ever even eaten a raw oyster? Is this a food people like?
I've never eaten.
No, and I think after...
that was my second. It's a 2-part question. Will you ever have one after this?
I have eaten one, but it was cooked and heavily doused in garlic and cheese. But I think, oysters eat the most disgusting food ever. Shellfish diet, I think, has traumatized all of that. That smell still haunts me. And so after smelling that, I'm good looking at oysters from a scientific side only.
I don't think it was even just what they eat. I think it was just like just extracting like over like 300 of them. Yeah. I think is what made me not want to eat any. I think at that point, I don't think I could ever eat a raw oyster, but not after
what
that.
We've done. No. Oysters are awesome. Appreciate them more. Thank you so much.
What's your favorite fun fact about oysters?
Oh my gosh, yes. Juvenile oysters are called spat, and I love that fact.
Good question.
I think mine is that they can filter up to like 15 liters of water an hour, I think. It's like a crazy amount. I'm not sure if that's like the specific amount, but it is a lot of water, and they do improve a lot of water quality, which is really interesting.
Yeah. Anybody else? Fun fact?
Oh, mine is that the oyster reefs can like, they can provide like various habitats like marine species, and also like the way how like they reduces like the wave energy and like stabilizes shorelines. That was kind of interesting to me.
Yeah.
I like, my favorite fact is like oysters are kind of like a keystone species. So like if they're taken away, many other organisms and marine life are heavily affected.
Yeah.
Mine is probably, it's not really a fun fact, but I remember seeing the oysters in the tanks and their little feet or
oh my gosh, those were so
like
cute.
crawling up. I don't really remember, but it was really cute. They're really cute.
Well, I'm hearing some conflicting things about oysters. They're adorable. They're disgusting. We love them. Thank you guys for joining us today for the Nerd Alert podcast. Thanks, everybody.
Thanks for having us.
All right.